Last time, I'm not the kind of person who's going to ask you to learn specific details on these chemical reactions. I'm going to get the general idea of these. So the general idea, as I draw up here kind of this deep into this topic, this is what I'm looking for. We basically have an RNA and it's got some three-dimensional shape. There's a soluble guanosine, or guanine, that comes in. They've got a ribose like this. We have the guanine poking off like this. This thing actually binds into a binding pocket in the three-dimensional shape of this thing. The 2' hydroxyl of this engages in a nucleophilic attack and breaks this covalent bond. There's now, this RNA has been broken into 2 different pieces. The shape of this is such that this 3' hydroxyl that's now free, so this is basically your phosphate group that got broken off there is now connected to this OH, right? Sorry, those of you at home can't see this. Let me draw this larger. So you have 3' hydroxyl here. There's the carbon up here, and then there may be a phosphate on there, I can't remember. So this 2' hydroxyl is now covalently connected with the phosphate that's connected on this 5'. This phosphate used to be connected to this hydroxyl, right? And the overall three-dimensional shape of this molecule positions this very close to some nucleotide. It's not just random, it's a very specific nucleotide based on the shape. And this engages in a nucleophilic attack that breaks this off. And the effect is we now connect, and the way we draw this when we show splicing, I should take it down, is we now connect this to this, like that. And this piece actually goes away. What we wind up with is this guy has a guanine nucleotide hanging on it in a kind of a weird linkage. Because this is a 2 to 5 connection instead of a 3 to 5 connection, right? And then we wind up with our spliced RNA where there was some specific splice site there. Okay? If you can describe your way through this process, or draw this very simple process, and I've gone in more detail just to make sure reviewing it and getting it in, that's what I'm looking for you to understand about splicing. That's it. It's more detail. If you want to study that, you can earn your PhD studying this process, okay? Doesn't sound fun to me. Any chemists in class who think that sounds like fun? Great. Have at it. I don't like it. Someone needs to study it. If you can understand this, group 1 splicing, group 2 splicing is really very simple. Group 2 splicing basically involves an RNA, and instead of a guanine coming in from outside, there's an adenine in here, actually in the RNA. And the adenine, basically there's a sugar associated with that, and that sugar is part of the sugar phosphate backbone of this, right? This sugar makes an attack here, which breaks this, which then can do an attack here. And what we wind up with, this is really interesting. Because this covalent linkage is gone over here, what we wind up with is, here we have our 3' hydroxyl here. That's this guy right here, that 3' hydroxyl. This goes around, and the 5' phosphate is connected to the 2' of the adenine here. You see that structure? It's a very interesting nucleic acid structure. We call this a lariat. Does anyone know what a lariat means? Has anyone ever heard that word lariat before?
A circle of flowers or something.
A circle of flowers?
Yeah.
Where have you heard of it, Sydney? You just heard it, you don't know what it means?
Well, like, I'm thinking of like the rope that they use, like a lariat rope.
A lariat rope, like a lasso?
Yeah.
Yeah, that's it.
Yeah.
That's what I've heard of that. A lariat is like a rope.
Fingers.
It's a loop,
And rodeo.
right? You've got this loop, and then you've got the cord pulling off of it. This is a loop right here. Okay? But basically the same thing. There's two nucleophilic attacks. You break the bump, you break the sugar phosphate backbone. That 3' hydroxyl then does a nucleophilic attack. And you wind up, this is your intron that goes away. And what you connect together, do that in red, what you stitch together is these two exons. This is exon 1, this is exon 2. Group 2 splicing can happen in a test tube all by itself. The RNA that you put in there folds up into a shape that it performs surgery on itself and cuts part of itself out. And for the purposes of this class, this is garbage which gets thrown away. What does that mean for those of you who've been in my class before? What does that mean? Huh?
Gonna say no for the exam is important?
No.
Nope, okay.
That means that it's not entirely correct what you're saying.
It means I just lied to you. For the purposes of this class, this is garbage and gets thrown away. People have gone and found these things that have functions. We're not going to discuss that. Okay? The wonderful thing about biology is if you can imagine it, if you can even think of something like how it might be used or that it could be used, some organism has done it. It's totally bizarre. That's why biology is so fun. Okay. I got to close out. I was running a grant when I came in here today, so a grant for teaching me how to brew beer. So you can imagine I was excited and got lost in it. So we are doing. So that's group 2 splicing. And group 3 splicing is basically the same thing. You wind up with a lariat structure. Group 3 splicing is almost all splicing that happens on Earth. And it can't happen. The RNA cannot do it on its own. It requires a big, huge machine to come in, and I'm going to expand the definition of machine in your mind. A big factory comes in and performs the work. Genetics, lecture 1, RNA processing. Yeah, that didn't work. How did we do this last time?
You were logging the other computer.
Oh, God. Murder. This isn't even on. Oh, there it is. It's going to take forever to load. Yeah. Oh boy. Okay, so I hope when I told you that the RNA folds up into the proper three-dimensional shape, such that it performs surgery on itself, I hope you're thinking to yourself, what are the odds that an RNA would fold up into that shape? It seems like that would be a really hard shape to fold into, and probably not a lot of sequences would be able to do that, right? There are very few group 1 and very few group 2 RNAs. Yeah.
How does it know where to do it? Like,
It's all chemical reaction. It's this guanosine binds in the right spot, and I'm making this totally up. I don't know the shapes of these things at all. I'm just going to make it up as an example. So the question, sorry, I got some feedback from the camera people here last time. The question was, how does a self-splicing RNA know where to splice? So if we have the RNA, and I'm just going to kind of draw some shape here. If the guanine binds in this spot right here, it positions the 2' hydroxyl at this spot, such that it will attack that spot. They're physically in three-dimensional space close to each other. And I need to draw more here. And now this 3' hydroxyl is in very close proximity to this sugar phosphate backbone, that it's going to attack that. And so you wind up breaking these guys apart so that this becomes the spliced RNA and goes away, and all the rest of this stuff is your intron. That makes sense? So it chooses where just based on the proximity of where the atoms are physically in space. That's it. So what I was getting at is how many sequences of RNA will fold in this exact shape? Not many. So if our RNAs in our genomes are going to splice, there's only a limited number of sequences that we could have in our genes that could be spliced. So most genes use type 3, group 3 splicing. The RNAs would fold into some shape that they would want to fold into. What happens is some proteins bind to the RNA and force it into this shape, or a different shape, that allows these reactions to occur. Does that make sense? So those proteins are called spliceosome factors. All right. Well, I hate Chrome. I want to do Firefox. I should get that off there. Oh, there it is. You really that slow today? Content, lectures, exam 1, they're all up here. Learning processing. Okay. So, other questions?
Is group 2 splicing always with adenine?
Yes, it's always with adenine. It's not, yeah, that's a
So is it also with the structure then? Because I mean, there could be thousands, millions of adenines and.
Yeah, so,
it's
yeah,
hard to
I
know
mean-
which adenine is.
Yeah,
just
so
like that
if it
one?
folds up, you've got, instead now you've got, there's an adenine that's in close proximity, and the adenine does it. But, there's an adenine up here, but it's not in the right three-dimensional space. So there's an adenine over here. It doesn't work.
So it's just where it's positioned.
Yep. And so the question was, is it always an adenine in group 2 that does the cut? I mean, yes. And we give that adenine a specific name. It's called the branch point adenine. Okay. Let's talk about group 3 introns. We'll get to this and we'll get to that later on today if we have time, I hope. So what we're going to do is we're going to define the 5' splice site and the 3' splice site. Understand how I got those? What we're going to do ultimately is we're going to cut across here. So this is intron. This is exon 2. This is exon 1. Because most RNAs cannot fold up into a shape where they are doing surgery on themselves, these proteins need to bind. And if proteins are going to bind, they need to have some specific sequence that they recognize. So we can define a consensus sequence. Now you understand why I was harping on a consensus sequence so early on in this course. For the entire semester, we are going to be talking about consensus sequence and consensus sequence and consensus sequence, right? Because I am such a nerd, I know the consensus sequence that these proteins bind to. The sequence right here is CAG, you don't need to know this, You can look it up. I've got it in the PowerPoint files and stuff. The sequence is C, oops, that's G. C, A, G, G, T, R, A, G, T, C, A, G, G, R. What is R? What nucleotide is R?
Just random.
Random? No, random would be N, for nucleotide. Good guess. Purine. What is so that's single letter code for purine. What's single letter code for pyrimidine? Y. What nucleotides are purines?
Guanine and
Huh?
adenine.
Guanine and adenine, yes. Of course, you guys know that, right? So, R is adenine and guanine. And when I asked, I had to stop and use my little mnemonic for learning. This is the other two then. That's the only one that spells a word. R-A-G.
Or you can do cut the pie.
What?
Cut the pie for pyrimidine or purine.
Cut, I suppose, yeah.
It works.
Also,
It
you're-
If it works for you, it works.
does.
Probably everyone in class is going to remember yours before more than they
Oh, right, makes a lot of sense.
Yeah. Okay. So what this means is this that, and this is what you'll actually. This is our next activity in lab. If you want, you can do this ahead of time. You can come in, and lab will be like a total breeze. You'll be playing your video game and half listening to me, right? We're going to now go. One of the reasons we found the locus of your gene last time, it seemed kind of useless at the time. You are going to go into your locus and you're going to get the actual DNA sequence of the chromosome where your gene is. And you're going to pull that whole thing out and you're going to find these in the genome and you're going to see, do I have consensus splice sites? And just like we did with the with Marilyn Kozak's sequence, you got the ATG and there was a sequence around it, and I said, if you don't have an A here, you've got to have a G here. Remember I said that? If you don't have it, if you're going to splice, man, you really got to have this and pretty much that. And you probably need 3 of those 4. These are nice. Those help. But this is critical. Now I want, especially those of you at home, I want you to notice, I said I'm going to splice here and I'm going to splice here. So this stuff is going to get cut out. Which nucleotide, what nucleotide is the 5' end of the intron once it's floating out in space there? What is that nucleotide? A, T, G, or C? G. Tell me when I get to it. That's it. Where's this G? It's
Oh,
the
it's in the center.
It's in the middle of the spliced RNA. What is it covalently connected to? A and G. This G right here? No. Which G? The next G, this G right here? No, this G. Right? All of this, for our purposes, is going to get cut out and thrown away. Yes? I'm going to end up with G, T, R, A, G, T, intron stuff, C, A, G, and then this G will be covalently connected to that because this doesn't exist. Now the truth is we're going to form a lariat structure here with branch point adenine. Okay? So, usually down towards this end, there's a branch point adenine. And this branch point adenine does the break here, and then this 3' end comes in and attacks here, and off we go. And usually, you'll have 3 or 4 or 4 or 4 of these, and you'll usually have 3 or 4 or 4 of the 4 of those. So, let me ask you guys this question. If I have a messenger RNA, and I told you this messenger RNA has already been spliced, how would you find the spot where it's been spliced? I'm giving you this sequence only. Using that sequence, how could you try to make a guess as to where it had been spliced?
Look for a CHG.
Yeah, use your find, Control F, and search for CAGG. And you'll find that, it'll be all over here. If I said it's only been spliced once, you'll find that sequence in here a couple times. But you can guess probably the spot it was spliced was one of these, and the intron was removed from there. Does that make sense? Good, okay, great. The reason I'm taking so much time to explain this is last year I didn't do a very good job, and nobody understood splicing. It was very clear from the exam. I was like, I just totally failed on that one. I'm not doing that again next year. I'll make sure everyone gets it. Okay?
So what exactly is the difference between group 2 and group 3 splicing? Because is that group 3 splicing what you just explained?
Yep.
Because it looked just like group 2 over there.
Chemically, it's exactly the same, but it cannot happen on its own.
Do you need like something to push it?
You need
like a factory?
You need a factory. You need a machine. And let me show you the machine. So the question was, how does group 2 and group 3 differ? Chemically, they're the same. But how it gets done is different. So, turns out, you have a machine, and I'm going to guess it's 6 proteins. And those 6 proteins recognize this sequence, and they come down and they bind, and that machine is called a SNRP. Small nuclear ribonucleoprotein particle, something like that. Things inside the nucleus that have what? Have RNA and proteins in them, and they make this big machine. So that thing binds there. We have a different SNRP. Binds here, and then we have even yet another SNRP that binds on the branch point A. So this is probably 6 proteins, this is probably 4, this is probably 6. And then what these guys do is this guy interacts with this guy and that guy and brings them all together such that these guys, these nucleotides here, are all brought in very, very close proximity and crammed right next to each other so that covalent bonds start breaking and such. Splicing happens. Okay? I won't go into details, but once this thing all assembles, some of the proteins go away, some other proteins come in, part of the reaction happens, some proteins go away, other proteins come in, more of the reaction happens, the reaction finishes, and most of these guys go away, but we're left with a couple of little proteins hanging on in different spots. The details of all that I don't know. But we have, basically what we wind up with is a spliced product with some proteins hanging on it. All right, let me see what I've got in this microscope, or in the PowerPoint file here. Okay, here we go. Here's the, let's see if I got it right. Here they actually give the percents for each of the nucleotides in the consensus sequence. And I said CAG, and they say it could be either an A or a C, and it's a 70% chance. And here they say it's a 100% chance that you have a G right here. They said 100% chance that this is a G. And they say it's a 100% chance that it's a T, U because it's RNA world, right? I wrote T because I think DNA world. And that's 95% chance it's an R, A, G, T. So I was right. I had my consistency was right. So they give the percents. So these proteins that bind, how do they find that consensus sequence? That complex of proteins that I drew? SNRP, it actually has its own little RNA. And we call it a guide RNA. So what the SNRP is doing is it takes the guide RNA, and this guide RNA has a specific sequence that is complementary to this sequence. So the SNRP is floating around the cell saying, hey, are you complementary to my RNA sequence? No. Are you complementary to my RNA sequence? No. Are you complementary to my RNA sequence? No. How about here? No. How about here? Oh, perfect, great. This is where apparently I'm supposed to splice. There's a guide RNA that hybridizes onto the branch point adenine. There's a guide RNA that's going to hybridize onto the CAG down here. So they've actually tested this stuff out. If I make a mutation in the guide RNA, and I make a similar mutation in the messenger RNA, does it still splice? Yes. Because the guide RNA can find, because the mutations match. Does that make sense? That's more than you need to know. These machines, these SNRPs, are so big, you can see them with an electron microscope. They can actually get three-dimensional shapes of these things. Such big, big machines that do the splicing. Any questions on group 3 splicing? Again, I don't want you to learn chemical reactions, I want you to get the general understanding. If I gave you a sequence, you could tell me what's in the intron and what's in the exon. Right? Separate fundamental understandings. You guys do not look excited at all.
It's Friday.
It's Friday. So the lab immediately next to me, one of my best friends in grad school, he studied, remember I said the reason I know, like 6 to 8 proteins bind and then reactions start and some things go away and some things come back. The reason I know that is because Jeff showed that. I was like, why would you do that as a PhD? That sounds really boring. Sorry, Jeff. Okay. And here they just show the reaction. And Jeff actually was the one who basically showed these guys, U1 and U4 go away. It's kind of cool. He showed these reactions. This figure is because of Jeff. How cool is that? Okay. So, let's talk about the importance of splicing. This is a gene called dystrophin. If you look on the genome, remember in lab, I was like, zoom out, let's see how much of your chromosome your gene takes up. Someone have one that was like 40,000 base pairs? Who had one longer than 40,000? Anyone? So 40,000 nucleotides, and then that thing got spliced down to how much? 2,500?
Yeah, something like that.
So 37,500 bases were cut out as intron and thrown away. The pre-RNA for dystrophin gene is 2.5 million base pairs. It is like 10% of a chromosome. That gene takes up that much space. So literally, RNA polymerase binds to the promoter, measures out, here's nucleotide 1, it makes a couple of abortive transcripts, and then it starts making the transcript, and a day later, it finishes. It takes 24 hours to transcribe the pre-RNA for this gene. And then the gene is spliced down to 14,000 base pairs. 2.5 million base pairs get cut out and thrown away. That's 2.5 million ATP molecules of value. More than that, because remember it's a pyrophosphate that comes off, so it's 2.4 million ATPs times 2. So you have to put both of those phosphates back on. Why? How can that happen? Why would this happen? Splicing must be really important to waste that much ATP, right? The organism has to get that kind of benefit out. And what is the benefit? We talked about that. another cliffhanger for you. We'll talk about that maybe in session, final section of the course. So, we started drawing maps out, and I wonder if I want to do that now. I've got 10 minutes. Okay, so let's do that. So if I have my DNA, you might want to get your pen and paper out for this. I have my DNA. I can make my pre-RNA. First step in the central dogma, right? So, what features can we draw on the DNA based on this little green line I've drawn? What features did we talk about in the DNA? If I know this, what can I say about this? Where's +1?
Right where the line starts.
Straight up here, right? So, and the arrow goes this way, right? Okay.
The promoter is upstream.
The promoter is upstream. Okay? What else can I do? One more that I can think of.
Termination
The terminator.
is to the right of the
Gonna get
RNA.
you. Come with me if you want to live. Do you guys even know what I'm talking about? All right, so far I think that's all I can do. All right? Am I missing anything here? No, great, okay. Let's go to the next step. Let's go to so this is transcription. So let's go to RNA processing. So I'm going to actually, so this is my mRNA. And now I'm going to draw the same thing, but right below it, okay? Actually, that's sorry. Okay. So let's use this. How can I mark this? Based on what I see here,
Introns and exons.
I can mark the introns and the exons. So I can say, I can mark my intron-exon boundaries. And I can actually say, I can go like this. So this is intron. This is intron. This is intron. This is exon 1. This is exon 2, exon 3, exon 4. Right? This is the 7-methylguanine cap. We haven't gotten to that yet. Don't worry about it. This is the poly A tail. You should basically know what that is. Anything else we can draw on here? One more thing I can see. what I got. I got one more.
Could put an adenine in each one of the introns.
Oh, I could put the branch point adenine in. I don't know where it is, but yeah, it's usually down on this side. I can put a branch point adenine in. Let's just do a circle so we don't mistake it as a splicing boundary. These are not the same length. How did that get cut? AATA. So that's the poly A signal. Does that make sense? Right? So now, based on how we've illustrated on here, what changes can I do to this illustration here to show this stuff on here? Why don't we do this? Looking familiar? See that? Who needs more time? Any questions? Okay, what's the next step I need to go? What's the next step I get to do?
Why did you erase this and just draw a line?
Because I'm noting where the
Okay.
introns and the exons are on this one figure of DNA. Okay, yes? I'm going to do this a little bit. Something like that. Pardon me, what's the next step in our central dogma? Translation. I haven't done red. I have my protein, my amino acids. This is the N-terminus of the protein. That's the C-terminus of the protein. Okay? What have I learned here that I can put on here? I can say this is the coding part of this, right? Yes?
What does that N and C stand for? Sorry.
The N-terminus and a C-terminus. An amino acid is this.
Oh, so like the amino side and.
Yes.
Okay.
Okay. The question is, what does the N mean and the C mean? N-terminus, C-terminus, the amino side, the acid, the carboxyl side of the protein. All right, you see how I did that under there? How can I put this onto here? So obviously this thing starts here, right? And it goes into there. Is this thing block or a line? Block. Is this thing block or line? Block. Okay, and then, so that's this spot is here. This guy, so this is this, this is this, this is this. So now I need to do this on here. Right? How do I put this onto here? Getting complicated? So this is going to be here, right? And then this is a box. Yep, this is this guy right here. This guy is apparently a box. So this is actually pretty easy. And this just goes up like this, right? Let's just say we'll go straight up on that. Good thing this video is going to be online, right? You can watch this again. How do I illustrate this on here? More boxes? I can do this.
Or just shake it.
What nucleotide is this first one right here? A, because it's part of codon is what? ATG, yeah. Right? That's the initiation codon for this protein that gets made. So this must be the termination codon, which is TGA, TAG, or TAA. There's 3 opportunities for it there. Does this look like what you saw in lab? So this is an incredibly informative figure. This figure tells me the structure and important things of the DNA. The important step in the processing of the RNA is illustrated by the splicing here. And it shows me what part of this is amino acid sequence. In a paper, most people who publish a paper where they say, oh, I'm studying the such and such gene, they'll put this up, and you are expected to be able to do this in 3 seconds in your head, because it's just a part of who you are. And my goal this semester is that you'll be able to do that. When I put one of these on the exam, you'll automatically know what these things are. The challenge will be, is this dealing with transcription or is it translation? Let's work through that. That's my job this semester, is to help work through that. Sorry, thanks for staying 2 minutes late. Sorry, I was late. And I will see you guys Monday. We're going to run through this multiple times. I need you to work with me though, okay?